Today we have Adilson talking to us. He's from the University of Melbourne in their Department of Surgery. He has a BSc and MSc in Biology from Rio de Janeiro State University in Brazil. And for his PhD, he uses biochemistry tools and performs experiments both in vitro and in vivo to investigate the role of EVs on cancer metastasis. Adilson has publications on a range of topics, including anti-TGF-beta therapies in clinical cancer therapies and why they might not be succeeding, as well as low-power lasers altering mRNA from genes in cancers, muscle injuries, lung injuries, and E. coli. So a plethora and a wealth of experience was all to draw on today.
Thank you so much, Will. Thank you, everyone, for having me here. As Will introduced me, my name is Adilson Teixeira. Today, we'll be presenting a paper recently published by the Only One group from the University of Cincinnati in the US. And the paper is entitled Autophagy Blockade Limits HER2 Breast Cancer Tumorigenesis by Perturbing HER2 Trafficking and Promoting Release via Small Extracellular Vesicles. So the authors introduce the main theme of the paper, starting talking about a little bit of breast cancer. It's the most common malignancy for women, and there is a different subtypes actually of breast cancer that are particularly characterized by molecular alterations, which include the alterations in the HER2 subtype with a driving force that is the amplification of the HER2 gene, affecting about 20-25% of all breast cancers. Thanks enough for the new therapies that have been developed in the last years. We have very good HER2 therapies that greatly improved patient outcome, including monoclonal antibodies that target specifically HER2, but also dual kinase inhibitors that simultaneously target HER2 and EGFR. Still, while these therapies are, as I mentioned, a great advance for the treatment of these patients, the acquired resistance to these therapies and also the recurrence of these tumors limit quite a bit the use of this treatment. So just to have a very brief overview, HER2 is a transmembrane receptor. It's a member of the ErbB kinase receptor family, and to have the activation of a signaling pathway downstream, this receptor has to dimerize with another receptor, another plasma membrane receptor, which can be such as exemplified here, member of the EGFR family. After the dimerization, we have then the phosphorylation activation of downstream signaling pathways, exemplified here, that has half MAPK, but also the PI3 kinase and the AKT. I will call your attention here for the AKT because it will be used by the authors in this paper as a way to determine if the HER2 is indeed activating downstream effectors or not. And as I mentioned, the HER2 signaling pathway is indeed not the problem itself. It's not the question itself in the paper, but actually it's to understand why the resistance to anti-HER2 therapies emerge in breast cancer patients. And one of the possible explanations, one of the recent possible explanations for that, it's the role of autophagy. Autophagy is a very tightly regulated mechanism for the clearance of defective proteins and also organelles. We have in this illustration here, and I think it's quite intuitive, it's quite straightforward to observe that we have a very large number of steps, regulatory steps, controlling the biogenesis of an autophagosome. And all of that starts when the mTOR is inhibited, and then we have this signaling cascade involving different protein complex. I will not go into details here. Actually, what I would like to show you is that through the activation of these signaling pathways, we have the nucleation of a phagophore, the elongation of this phagophore to englobe defective proteins, as I mentioned, or organelles, and then the formation of an autophagosome. And the main target of this project, of this particular work, it's probably FIP200. We can observe it's involved in a quite early step for the activation of autophagy. But throughout the paper, the authors will also investigate and use as a confirmation for the results the detection or the knockdown or knockout of other proteins, including ATG13, also ATG5, and LC3, which is a common marker for autophagy. And as I mentioned, autophagy is a very regulated process, and it is involved in multiple scenarios of health and disease. Autophagy has been described to play roles in multiple types of cancer and of course non-cancer disease as well, but particularly in cancer, it has a contradictory role, and both anti and pro-tumorigenic roles have been described and related with autophagy. As you can see here in this figure, this is a figure from Wei et al. 2011. It's a publication from the same group earlier in 2011, and they used a mouse model of breast cancer, MMTV PIMT, and generated a knockout model for the gene FIP200, a fluxed model for these animals. And we can see here that while control animals or even heterozygote animals represented in blue and red respectively had a relatively low tumor-free survival, animals showing the knockout, exhibiting a knockout for FIP200, which would mean an impaired autophagy, present a very delayed formation of tumors, development of new tumors, and of course, as a consequence, increased tumor-free survival, which is also reflecting an overall increased survival for animals with impaired autophagy. And it's quite interesting to note that low autophagy gene signature has been also correlated with HER2 breast cancers, and it has been described for these patients for these types of tumors. But as I mentioned, opposite roles have been described for autophagy, and it's not exactly clear so far if these pro-tumorigenic roles or anti-tumorigenic roles are exactly related to the classic function of these genes and proteins encoded by these genes associated with autophagy, maybe with alternative functions. And among these alternative functions could be an interference, could exist an interference with the trafficking of other intracellular vesicles, other vacuoles that could, for instance, result in the secretion of small extracellular vesicles. A large population of small extracellular vesicles are the exosomes, and it's being postulated that autophagy genes and proteins encoded by these genes can regulate secretory pathways, including the secretion of small extracellular vesicles. We can see here in this illustration that the exosomal pathway starts downstream the endocytosis, but instead of following the lysosomal pathway, endosomes will follow the exosomal pathway to form multivesicular barriers or MVBs, and inside these multivesicular barriers will be released interluminal vesicles or ILVs, thanks to the activity of several protein complexes, including the activity of Rab enzymes and snares, and also the cytoskeleton and several others. You have the transport of these MVBs, the docking and the fusion of MVBs with the plasma membrane, and then the interluminal vesicles are released as exosomes. On the top of the figure, we can just highlight here how different other protein complexes are required for the sorting of protein cargo into exosomes, and also the biogenesis of multivesicular barriers and ILVs. And because these small extracellular vesicles, with particular focus here on exosomes, after they are released, they have been considered as major mediators in cell-to-cell communication. The authors then hypothesized that by downregulating or by impairing autophagy in breast cancer cells, it is possible maybe to impact the secretion, the biogenesis and secretion of these small extracellular vesicles as well. Therefore, raising at least three important questions. One is, what is the role played by autophagy during breast tumorigenesis and progression? If there is some role played here, do the mechanism regulating autophagy in some way crosstalk with the biogenesis and secretion of these small extracellular vesicles in this particular context? And can the small extracellular vesicles secretion impair some way the breast tumorigenesis in cancers that will naturally exhibit, or forcibly in this model exhibit, reduced and impaired autophagy. So to answer these questions, the authors will be mainly using two different models, an in vivo model and an in vitro model. The in vivo model is slightly different from the one I showed you before in the publication from the same group. Here they will be using a mouse model, an MMTV new mouse model. This model carries amplification of the HER2 gene, therefore recapitulating what is observed in breast cancer, in human breast cancers at some extent. And based on the Cree recombinase system, in the use of a Cree recombinase system in these animals, they will generate a knockout mice for the FIP200, impairing autophagy, or a knocking model. But in this case, FIP200, it's actually expressed as a dominant negative version, a dominant negative isoform that cannot interact with ATG13, and therefore result, or at least it is expected to result in a similar phenotype with impaired autophagy. And the results obtaining in knockout and knocking animals will then be compared with control animals with our heterozygous or wild type for FIP200 and expected to exhibit a regular autophagy. While this in vivo model is, of course, quite important to understand the phenomenon as a whole in a scenario that's more closely recapitulate the tumorigenesis in human patients, there are some limitations, and to overcome these limitations, they isolate some tumor cells that will be spontaneously immortalized, and FIP200 fluxed tumor cells from MMTV new mice, also again using the Cree recombinase system, but now sensitive to tamoxifen, will then generate two cell lines. They are overall the same, present the same characteristics, which is the N148 and N418. In the absence of tamoxifen, they will remain as a wild type phenotype, but if treated with tamoxifen, they will be knocked out for this gene, therefore with exhibiting reduced autophagy as well. So the first question is, getting back to the previous slide, how autophagy can modulate or interact or somehow impact breast tumorigenesis in the HER2 amplification context. You can see in the top left in figure 1A, that control mice, wild type for FIP200, exhibit at least 50% of the group develop tumors around 40 to 50 weeks. And by the end of 70 weeks, pretty much all of them will have developed tumors. But when the FIP200 is knocked out, represented by the red line, or even the knocking model, this tumorigenesis is dramatically impaired. As we can see here, a very minimal proportion of these animals will develop tumors, and this will happen very, very late throughout the process, increasing, therefore, the tumor-free survival of these animals when in the scenario where they're presenting impaired autophagy. In agreement with this result, we can see here on figure B that the knockout, when tumors are isolated from these animals, and then the protein lysate is investigated by immunoblot, we can see that the knockout indeed works, and not only that, but the knockout of FIP200 in these animals increase the abundance, the levels of P62, showing a decreased level of autophagy, and on the other hand, decrease the levels of phospho-AKT, demonstrating that by knocking out FIP200, some way you have the downregulation or the inhibition of the downstream effectors, or of effectors that are downstream HER2. You can see here on the figure C by histologic analysis that control animals exhibit, after a while, this morphology in their mammary glands that are typically lobulo-alveolar morphology, which is not observed in the knockout and knock-in mice for FIP200. And in addition, while control animals exhibit higher levels of Ki67, which is not observed in knockout and knock-in animals, they exhibit lower levels of P62 as previously determined by immunoblots, once more confirming that the knockout or the knock-in in this case for the FIP200 not only result in reduced proliferation of the cells in the mammary gland of these animals, but also in reduced autophagy. We can see here on figure F that after 30 weeks, but particularly after 50 weeks, control animals exhibit increased levels of HER2 in the mammary gland and in tumors in this case, which is not observed in knockout and knock-in animals. And not only that, after 63 weeks, these higher levels of HER2 are associated with decreased levels of P62 and increased levels of phospholipid A in the inverse of what is observed here for knockout mice. So from these results, you can conclude that FIP200 ablation essentially will block mammary tumorigenesis in this model of MMTV neo mice. Still, as addressed by the authors, there is a limitation that is in a way mechanistically and another way statistically. Very few knockout and knock-in animal will develop tumors, so therefore it's different, or sorry, it's hard to compare what it's happening in terms of biological mechanisms in tumors developed by knockout and knock-in mice with the control mice. It's hard to precise the mechanisms, and to overcome this limitation, which is in a way statistically statistical limitation, authors then developed, as I mentioned before, an in vitro model isolating tumor cells from these mice and generating by using a fluxed FIP200, generating a knockout in these cells by treatment with tamoxifen. We can see here in the figure B and C that both cell lines, NE148 and also NE418, will exhibit reduced levels or mostly absent levels of FIP200, demonstrating the deficiency of the knockout in association with lower levels of HER2, of total HER2 in the cells, similar to what was observed in vivo. And furthermore, when the cells are grown in starvation conditions, particularly when they are treated with BAFF-A1, which is an autophagy inhibitor, they exhibit lower levels of LC3B compared to wild type cells, demonstrating reduced levels of autophagy as well. More than that, knockout cells represented by the red line show reduced proliferation when compared to wild type cells and also reduced migration. And when these cells, wild type or knockout, are implanted in wild type animals, in wild type mice, FBV mice, we can observe that the tumorigenesis in animals implanted with wild type cells, it's quite abrupt. And after 30 weeks, we have almost all these animals, or all these animals actually, have developed tumors, which is delayed when these animals are implanted with FIP200 knockout cells. Not only in terms of tumor-free survival, there is a benefit in this scenario, but also tumor volume and total tumor weight is reduced in animals implanted in tumors formed in animals that were implanted with cells FIP200 knockout. And of course, so far, what the authors have clearly demonstrated is that FIP200 exhibit a clear role in breast cancer tumorigenesis in this model. But so far, how to determine if this is specific for FIP200 or this is more broadly associated with autophagy? We can observe here in figure G that to overcome this limitation, once more, the authors used a short herping RNA targeting a different gene acting downstream in the autophagy pathway, which is ATG5, as I previously showed you. Both cell lines using different short herping RNAs exhibit reduced levels of the target gene, but also reduced levels of HER2. And not only that, but not only for these tumor cells isolated from mice, when HeLa cells were submitted to a CRISPR-Cas9 knockout model targeting FIP200 or third different gene associated with autophagy, that is ATG13. Not only, again, the knockout result in successfully result in lower levels of the target proteins, but also reduce HER2 levels in these cells. And once more, when HeLa cells are cultured in starvation and treated with BAFF-A1, they exhibit lower levels of LC3B, particularly in the second band here, compared to the wild type, once more demonstrating impaired autophagy. And in the same way, they have very well explored genetic mechanism to impair, to block, to reduce autophagy. We can see here in figure K, they were very successful in demonstrating that this can also be obtained by a pharmacological treatment with different types of inhibitors targeting autophagy, leading to the same pattern of result, which is impaired, lower levels, lower HER2 levels in these cells. And to finalize this panel, you can see here in figure L that this is not limited to epithelial cells or cells with epithelial origin. In figure L, authors have used mice embryonic fibroblasts and knock out these cells for 50 to 100. And once more, with a successful knockout, they also demonstrated reduced levels of HER2. So from these results, you can once more conclude that HER2 levels are reduced in cells with impaired autophagy. This is not limited to 50 to 100, but it's really seems to be associated with autophagy as a whole. And these results, they now obtained in vitro, clearly recapitulate what was previously demonstrated in the figure 1 in vivo. But this actually raises a question, that is, if HER2 is being downregulated in this context, what is triggering this downregulation? The first question would be to understand how the half-life of HER2 is impacted in these different contexts. So we can see here in the figure A and figure B that when wild-type cells and knockout cells, tumor cells isolated from the tumor of MMTV mice, were treated with cyclohexamide, which is a potent inhibitor of protein synthesis, the half-life of HER2 is decreased in knockout cells compared to wild types. You can see here in the graph the same pattern being shown. But it's quite interesting, and you can see here in figures C and D, that when the cells are treated now with MG132, which is to inhibit protein degradation, but proteasoma degradation in this case, although we can see some increasing in HER2 levels, this not rescue the levels that were previously observed in wild type cells. It's not rescue to the same level. This is true for tumor cells isolated from mice, but also is true for HeLa cells knockout for 50 to 100 or knockout for ATG13. We can observe, or we can at least understand from these results that there is a reduction in HER2 levels, but this doesn't seem to be related, or at least doesn't seem to be mostly related with protein degradation. So what exactly is triggering this downregulation? To confirm the previous results, the authors then will decided to analyze by immunofluorescence the mammary gland of this animal's control or knockout or knocking for 50 to 100. You can observe here in figure C that cells from the mammary gland of control mice clearly exhibit a strong staining in the plasma membrane for HER2. This is reduced in knockout and knocking mice, although some cytoplasmic staining is still observed. And this is not only observed by immunofluorescence, but as we can see here in figure F, the same evaluation was carried on by FACS, and similar results were obtained, and the HER2 levels are indeed reduced in the plasma membrane of 50 to 100 knockout animals, or cells of 50 to 100 knockout animals. And this is not a generalistic result. We can observe that for epithelial markers such as EpiCon and CD24, there's this decrease is not observed. Actually, if something, there is still a slightly increase in level of these proteins. This is, again, not only true for tumor cells, but from tumor cells from in vivo models, but when these cells are isolated and cultivated and then examined in vitro, we can observe here on figure G that wild type cells exhibit the typical localization of HER2 in the plasma membrane that is decreased in knockout cells with a strong staining in the cytoplasm, and HeLa cells exhibit exactly the same pattern with a strong staining in the plasma membrane for wild types that is reduced in FIP200 and ATG13 knockout. So we can understand from these results that autophagy deficiency reduce HER2 localization at the plasma membrane, but there is still a question that is, if this protein is not being degraded, or at least not, this is not the main mechanism to reduce its levels, and this protein is not either, it's neither being transported to the plasma membrane. So what is the destination of HER2 in these cells with impaired autophagy? We have previously described that altering autophagy can possibly affect the intracellular trafficking in these cells. So one of the possibilities would be to interact by cross-talking with endocytosis and with the biogenesis of MVBs, alter the pattern of HER2, of intracellular HER2. You can see here in the figure A that when these cells were stainned for HER2 and Reb7, which is a typical marker for late endosomes, while wild type cells hardly, in wild type cells, we will hardly observe any colocalization. This is quite easily observed in knockout cells, demonstrating or at least suggesting that HER2 is being trapped or hence the localization of HER2 is being increased in these late endosomes. And this is also true in HeLa cells when, by using a fusion system, the GFP HER2 colocalizes with mCherry Reb7, but particularly this colocalization particularly happens in knockout cells, but not so much in wild type cells. And more than that, again, in HeLa cells, when the GFP HER2 is analyzed, now in cells overexpressing a mutant form of Reb5, which is a mutant form that pretty much blocks the maturation of early endosomes into late endosomes and therefore impact the maturation of multivesicular bodies, we can observe once more the colocalization of HER2 and mCherry Reb5 in knockout mice, but again, not so much in wild type mice. So this is quite suggestive, demonstrates that HER2 is being encapsulated, may not be the best term, but is indeed being accumulated in endosomes. But still, there is a question about what exactly is resulting in these higher levels of HER2 in endosomes. It could be as a possibility that these cells knock out for FIP200 or anyway with impaired autophagy would exhibit increased endocytosis, higher levels of endocytosis. And to evaluate this, we can see here in the figure B and figure C that the authors decided to evaluate the endocytosis by staining the cells with HER2 and using live cells follow HER2 after 3 hours. It's quite clear here on the top left that at the start of this experiment, wild type cells show the typical localization of HER2 in the plasma membrane, which is decreased in knockout cells, as we already saw this in the previous figures. But after 3 hours, as observed by the number of red puncta representing the cytoplasmic HER2, the levels of cytoplasmic HER2 are pretty much the same through all these conditions. So this actually shows that there is no increase in the endocytosis. At least it doesn't seem to be observed, this increase in endocytosis in FIP200 knockout or ATG13 knockout cells. And pharmacologically, when the cells, when HeLa cells, wild type or knockout, are treated with dinosaur to inhibit endocytosis, this not really rescue HER2 levels, not even closely to what was observed to wild type cells. So apparently there is no alteration in endocytosis itself, but the trafficking of HER2 in the cells, it's being different. It is different. So why exactly it is different? We can see here in the figure D that when the GFP HER2 is observed or is investigated, localization of this protein is investigated, it colocalizes with the Golgi marker, Golgi97 in red. But again, particularly in knockout cells, but not so much in wild type cells. And this also happens in HeLa cells, again, but now with a different marker for the Golgi. So there is an alteration in the trafficking of HER2. It's still increased in Golgi, but it's not being trafficked to the plasma membrane. Apparently, it's being, again, as I said before, it's being entrapped somehow in the endosomes. So HER2 trafficking from Golgi to the plasma membrane is diverted to endosomes in cells with impaired autophagy, but we don't know exactly why this is happening or how this is happening. And the question is, if this accumulation is happening in the endosomes, and considering that endosomes can originate or are part of the biogenesis of extracellular vesicles, small extracellular vesicles, and exosomes, could this accumulation of HER2 in endosomes lead to increased secretion of HER2 through these extracellular vesicles? So to initially investigate, we have to remember that before we have exosomes, secreted exosomes, MVBs show interluminal vesicles as, in a way, precursors of these exosomes. And we can see here in the figure A that when the endogenous HER2 is extended in the mammary gland of control knockout or knocking mice and co-extended with the anti-CD8 endogenous CD8-1, which is usually used to mark interluminal vesicles, there is a co-staining in these cells of the mammary gland of knockout and also knocking mice, but this is hardly observed in control mice. And when the cells are isolated, we can see here in the figure B that wild type cells, the colocalization of HER2, endogenous HER2 in wild type cells with a different marker for interluminal vesicles, that is CD63, it's not seen or not commonly seen, but increased in FIP200 knockout cells. But this shows us that it happens in animals, in mammary glands, it happens in isolated cells. Now in figure C and D, we can see that this also happens in HeLa cells, in knockout cells for FIP200 and ATG13. The colocalization of HER2 and CD63 or the red fluorescent protein fused to CD8-1 is observed in cells with impaired metastasis. But we still don't know exactly if the colocalization of HER2 in interluminal vesicles will indeed lead to higher levels of HER2 in the secreted extracellular vesicles. Although this is an important step of demonstration, it's important to conclude this investigation. And we can see here in the figure A, B, and also in the supplementary figure S5A, that when the small extracellular vesicles are isolated from the conditioned medium of wild type or knockout cells, the HER2 levels in the knockout cells, on the extracellular vesicles isolated from knockout cells, is indeed increased compared to wild type cells. But this is not exclusivity for HER2. We can also observe increased levels of ALEX. We can observe even an increased level of EGFR, and this most likely represent increased levels of total protein being secreted in the extracellular vesicles of knockout cells. Still, when these authors analyze the number of small extracellular vesicles in these conditions, although we can see some difference between both conditions, this difference is not statistically significant, representing that the number of small extracellular vesicles secreted by wild types or knockout cells is pretty much the same. And still very important, there is no major alteration in diameter of these extracellular vesicles secreted by both cell types, which leads us to the figure C here in the top right. When these small extracellular vesicles are, or these tracts of these extracellular vesicles are normalized by protein mass, and we have here similar bands for ALEX, we still observe higher levels of HER2 in the EVs secreted by knockout cells. This is also true for HeLa cells, as we can see here in figures D and E. Doesn't matter if normalized by protein mass or the number of SCVs, but this seems to be the case here, that in HeLa cells, knockout for FIP200 or ATG13, there is an increase in the levels of total and secreted small extracellular vesicles, although there is no alteration in the diameter of the vesicles secreted by these cells in any scenario. To further confirm that maybe this is indeed, for HeLa cells at least, related, at some extent at least, related with increased secretion of EVs. The authors then overexpress once more the RAB5, the mutant RAB5, to observe enlarged endosomes that cannot progress, and enlarged MVBs that cannot progress. And in knockout cells, they observe the colocalization of CD63, suggesting increased levels of interluminal vesicles in this in the multivesicular bodies of these cells, which is not observed in wild type cells, which again suggests us that in this scenario we have increased biogenesis of interluminal vesicles and possibly exosomes. And to finalize the paper and lead us to our discussion, the authors will conclusively determine the role of exosome secretion or small extracellular vesicle secretion in HER2. And this is quite interesting to observe that to do that, they use the siRNA targeting RAB27A to block the docking and the fusion of multivesicular bodies with the plasma membrane. And we can observe here that in knockout cells for FIP200 ATG13, that are also knocked down for RAB27, there is an increasing, or even we can use here the word rescue of HER2 levels compared to the wild type, showing that when we block the secretion of these exosomes, we can increase back again the levels of this protein, or at least the total levels of this protein. But it's quite interesting to note here in the supplement that although the total levels of HER2 are increased when we block the secretion of these EVs, there is no alteration in the staining of the HER2 at the plasma membrane in either of these conditions. And not only that, but in knockout cells for FIP200 ATG13, when the RAB27A is knocked down, there's no rescue of the of levels of phospho-AKT, showing that even that we have accumulation of HER2 inside the cells, this HER2 apparently, at least in these tested conditions, it's not activating downstream factors, which could mean that it's not really useful for the cells, at least in the way that was evaluated. So in conclusion, the authors propose in this paper that in cells with intact autophagy, HER2 is mainly trapped from the Golgi to the plasma membrane, and once reaching MVBs, you will fuse and colocalize, leading to the degradation of HER2 into lysosomes by autophagy. But when autophagy is impaired in these cells, in autophagy deficient cells, there is an alteration in the trafficking, and the HER2 that comes too early in the somes accumulated in MVBs will then be mainly secreted by the cells, and this will happen through small extracellular vesicles. So to finalize the presentation, I would like once more to thank you everyone that it's been present and here taking part of this discussion. I would like to thank you a lot, the student network on EVs and the International Society on EVs, and of course all the authors who took part and collaborate so this work could be produced. Otherwise, there will be no discussion here and about this paper. Thanks also my supervisor and all the members of the Cancer Signaling Research Laboratory. The discussion has been critical for my progress as a student, and invite us to everyone to follow our Twitter profile on cancer at Cancer Signaling. It's quite new, actually. We're still working on the first post, but you can follow us to not only know a little bit more of the progression of us students, but also share some ideas about cancer metastasis with a particular focus on TGF-beta signaling. Thank you so much. Stop sharing my screen.
Thank you very much for that, Adilson. That was a really, really nice presentation there, and you explained it all really nicely and clearly. So we have our first question, and while it's been asked, if I can encourage everyone just to post their questions in the group chat, and we can have sort of some more discussion. So, Sai, do you want to ask your first question, please?
Yes. Yeah, thank you so much for the talk. It's really fantastic work. It's very interesting to know a cytosolic protein that is controlling the plasma membrane one. So I have two questions. First one is, have you guys tried overexpressing FIP200 and study the phenotype? because upon knockout you saw the phenotype, but what about overexpression or?
Yeah, so Sai, this time this is probably a little bit different. So just to clear up, I'm not part of the group. This is just really a paper I selected to present. So from what I saw in the papers previously published by the authors and this one, no, I cannot. Sorry, this is the limitation I have. I cannot say what they are doing exactly in the lab. But I agree that it could be very interesting. Although I could raise a concern. I do not study autophagy, but in a way I could anticipate that maybe increasing, if overexpressing FIP200 would, which not necessarily happens, but would lead to a directly increase, a linear increase in autophagy in the cells. This could be detrimental for cell proliferation in a way. But autophagy, as I mentioned before, it's quite tricky. So it's hard to determine without, just by guessing, it's hard to determine the phenotype of the cells will be reversed as a whole or not. Maybe not. Maybe actually more proliferation could be observed.
Yeah, thank you. And I have a second question, because you said they have observed the phenotype of increase in the expression of small EVs. What about the metastasis? Because the field says that more EVs, more metastasis, at least in particular in breast cancer. So what is your thought about increase in SEVs and then metastasis phenotype? What would you comment on that one?
So I think this is actually a great question, but I shouldn't be using this word "great question" just because it's shared with my own opinion. I also thought exactly the same thing when I was trying to find some papers to discuss, to bring it here, and I found this one. This was my first thought. We have this tumorigenic model that we follow up to 7 weeks, apparently increasing the secretion of extracellular vesicles, and it's decreasing tumorigenesis. But then there are, I think, two important considerations to be keep in mind here. One is that this is not a model of exactly of cancer progression. So we don't have the evaluation of metastasis here, right? So this is a very important point. Once more, just guessing. I think that if this was a model aimed at evaluating metastasis, we may be seeing something a little bit different or entirely different. But the second consideration I have is that we have a very large and increasing body of evidence demonstrating that SEVs can transport this oncogenic phenotype from to other cells. But what I think is we still don't know exactly how it happens. After uptake, after you have the uptake of this small extracellular vesicle of extracellular vesicles, what is the next step? So far, I can see, and of course, please, anyone that can jump in and share these thoughts. The best we can track is what happens after we have these vesicles fused with lysosomes. But after this fusion, we could on the one hand think that the activity is gone. But what happened in between, I don't think it's quite clear. So maybe in some scenarios, if you have this, for instance, here we're talking about a transmembrane receptor. If it's being transported in the membrane of the vesicle, it could lead to one result. If it's transporting as an internal cargo in the lumen, maybe it will lead to a different result. Maybe the activation by interacting with extracellular proteins of components in the plasma membrane of a recipient cell leads to one thing, which could be different if you have the following to the lysosome. So sorry if I cannot give you a proper answer. I agree with you. I would expect to have increased tumorigenesis, or at least if we're following metastasis, increased metastasis. But I think it's quite good to see that might not be the case in every scenario we're studying.
Yeah. Thank you very much. Great talk.
Thank you, Sai.
Yep, nicely explained. Just in terms of time, I'm going to read this question out from Lilai. She's just asking for confirmation on the method used to isolate the small EVs and wondering if the HER2 is a soluble fraction, maybe?
Yeah, so there's, as much as I remember, of course, I could double check and even send the paper so everyone had access to the methodology. But as far as I remember, they used this differential centrifugation method, and to separate those typically 10K and 100K, and to separate this 100K fraction, there is no centrifugation, for instance, in gradient tubes or any other method, or isolation with specific antibodies or to trap. This is not the case. And in a way, it may even be a little short, the period of isolation for the 100K. Some protocols determine that it's hours or even overnight in some case, and here it's quite short. I think it's less than 1 hour actually.
Okay, brilliant, thank you. We've now got 2 questions from Lucia. If you could unmute and ask your questions, please.
Yes, hi. Excellent talk. Thank you very much. I have 2 questions. One, the last figure, if I understood correctly, it does show that the plasma membrane levels are comparable for HER2, correct? How do they explain it? We would have expected a change there.
Yeah, they don't go too much into details. One thing to consider is that in that last figure, this is a supplement figure. In that last figure, they're not evaluating, they're not comparing, at least it's not in the figure, they're not comparing with the wild type cells.
Okay.
Those 2 graphs are just for knockout for FIP200 and knockout for ATG13 HeLa cells.
Oh, okay. Thank you.
Plus minus Rep27A knockdown. So plus minus not Rep27A knockdown, there's no difference. But comparing it with wild type, probably those curves would be different because as long as everything we saw, right? Another consideration I think we have to keep in mind here is the timing. After you have the knockdown for Rep27, how long after that you evaluate HER2 at the plasma membrane? So you're blocking autophagy, so a huge source of degradation, it's not there anymore. You're blocking the secretion in this small EVs, so they cannot be released. If you probably incubate for long enough, if you keep this model for long enough, I would expect this to somehow lead to the plasma membrane. But this is just speculation. It may be, let's say, if it could be in. Once more, sorry if I don't like to guess in this case, but I think this might be the case. You could lead to a cytotoxic effect if you're trapping this protein that should not be there in this particular condition. You could lead to some cytotoxic. But once more, we don't have the data to see. It would be great if the authors could share some thoughts with us. Maybe they are evaluating this right now in the lab. It would be great.
Thank you, then. Bill,
Thanks
I
so
can
much.
skip my second. Thank you, Adilson. Very nice discussion. We can leave my second question out because and then you only have 10 more minutes, and I see more question coming in.
Oh,
all right, thank you so much, Lucia.
Yep. We now have a question from Guido. I apologize, everyone, if I do pronounce your name wrong, by the way, but if you could unmute and ask your question.
Lately, we have thanks for the nice presentation of that paper. But my question is, are we really dealing with what we called autophagy? Or are we dealing with non-canonical functions of factors that are well known in pathways of autophagy? Because I don't see immediately, if we start calling the endocytosis of a receptor in its turnover and its routing to exosomes or to lysosomes, that this is also autophagy. Ideas are getting confused, and so can you help me clarify that? Is it really, did they really investigate the relationship of HR2, I would say the basic conditions, to the normal mechanisms of autophagy? Because the use of chloroquine or bafilomycin it inhibits autophagy, but it inhibits lysosomes. It will also inhibit what would be considered as classical endocytosis lysosomal degradation pathways. I thought that autophagy was really linked to sequestering of cytosolic components and bring them to pathways that go into the lysosomes. That cytosolic contents and organelles. So help me, please. We're getting confused with all the the claims and the pathways.
Yeah, thank you so much, Guido. I'm as I mentioned, I'm not so much of an expert. Well, as a graduate student, I'm not so much of an expert in anything actually, but I'm not so much of an expert in autophagy. And I think your question makes all sense. And I think it's part of their question as well.
The way they, for example, LC3 is involved. Okay, it's lipidase, it's involved in autophagy. It's involved also in LC3-mediated endocytosis or phagocytosis. Last year, about a year ago, we saw that LC3 is involved, implicated in the loading of exosomes with RNA binding proteins and loading them with RNA. That's not the classical autophagy function. And so all what you have inhibited has to do with the making of LC3 as well, and it's lipidation. But
yeah, I agree.
Is it a function of autophagy? So maybe I'm a bit severe, but
yeah, I agree. I agree.
I cry for help because I'm getting confused. That's
yeah, I agree with your position. I agree with your questions. I have the same question. I think it's part of the questions the authors have, and unfortunately, I cannot give you an answer. I think the best way they try to address or to isolate autophagy as a process, it's by trying to target different components. But as you mentioned, these components are part of probably most broad process in the cells. And then the cytosis, it's related with these multiple steps. And you have some markers that were canonically associated with autophagy, like you mentioned for LC3B and the lipidation of LC3B. But now that we are having some new thoughts and some new evidence about the secretion of extracellular vesicles, I myself don't know. I don't have this answer. I don't think they try to. I think they try in a good way to isolate this effect, but I don't know if this is definitive. Maybe a future work of the same group could shed some light on that.
Honestly, I didn't understand the diagram and how the making a cross over the autophagosome is all of a sudden bringing the vesicles to the extracellular part. I can imagine if you would poison lysosomes and they get filled up with vesicles that normally should be degraded, ultimately you will create more vesicles. But are these the exosomes that we classically consider as exosomes? That's, I think, an unjustified conclusion, because if you look at specific cargo, I can tell you, you can use inhibitors of lysosomes, but that doesn't automatically yield more secretion of that cargo as vesicles. It's not a ubiquitous phenomenon.
Yeah, I think they avoid the word exosomes for that matter. We have very clear, I don't know if very clear, but I want to believe that very clear or the best we have for determining when to call exosome if you can see them on act being produced and being secreted. So they avoid the word exosomes. We can see in one graph of the supplements the word is there. Maybe was before for being edited for submission. I don't know. So we can see that it is there, but they call throughout the paper, they call small extracellular vesicles, because I think the best way they can determine what it is by judging by the diameter, and they use some markers as I showed you on the plots. But I think that the classification, it's mainly based on the diameter of these vesicles.
Thank you.
Thank you.
Thank you, Adilson. Very well dealt with all the questions. Thank you, everyone. It's been a really engaging discussion. I think we've all learned a lot. I mean, myself as sort of a cartilage biologist, it's been a rapid learning curve, but Adilson, you've explained it really clearly, really nicely. I think that is the end of the questions. I believe in Melbourne now it must be nearly midnight for you, so I think we might call that a day here, but thank you very much for that talk.
Thank you, Adilson. Thank you, Will. And thanks everybody for joining today. Let's do this again sometime soon.